Hello, my dear students. So today we are going to discuss a very important topic over that of fermentation. We are going to discuss about microbial spoilage as well as we are going to talk over that of the hurdle technology also, a special kind of preservation kind of mechanism which we are going to take care of. Okay. So we are going to focus today over that, like how the fermentation is getting done, what is the basic principle for that of the fermentation, as well as that of what are the different kind of that of the fermented products that can be produced, and what are the different enzymes, what are the sources from where the fermented products are being produced. All these things in a bit detail that we will be seeing it upon. Okay, so let's start about the same. First of all, we will be trying to know, first of all, we'll be trying to understand that what is fermentation. So if you are saying about the fermentation itself, generally fermentation, it is getting what I would say that fermentation, it is simply, I would say, the microbial cell propagation method. Okay. Now, what do you mean by microbial cell propagation? Propagation word, this actually stands for that of multiplication. Whenever we are using the word propagation, this propagation word, it stands for that of the multiplication itself. So whatever the propagation it is getting happened, so we are generating the products also. Okay, and that can be produced under that of the conditions like aerobic condition, like microaerobic condition, or anaerobic condition itself. In all these three different kind of conditions, the fermentation can be done. So what is this aerobic condition? If I'm saying about that of this aerobic condition itself, means ample amount of oxygen is remaining present, we know very well. What about the microaerobic condition? That means a limited supply of oxygen is been applied, a limited supply of air is been applied. And you can understand by that of the anaerobic condition, that stands for that of what? That means it is actually what? It is actually the kind of the fermentation which is occurring in an environment in which there is no oxygen, there is no air, there is no oxygen itself. That thing we are representing it as the anaerobic condition itself. That thing we are calling it as fermentation. So ultimately, when the fermentation is getting happened, what is the major use of fermentation? The major use of fermentation is to produce certain important kind of the components itself. And whatever the important kind of components are getting produced, those important components, they can be commercially exploited also, they can be economically exploited also, and they can be utilized for that of like medicinal approaches, medicinal extractions, and many of the other processes also, they can be utilized itself. Okay. So if we are saying like what is the biochemical as well as industrial meaning of the fermentation? So when like fermentation processes, they can be also used in the biotechnological processes also. Fermentation can be used in the alcohol industries also. Even the fermentation, it can be also like I would say that can be also considered under that of what? Can be also considered under that of I would say like dairy industries also, bakery industries also, and all the different kind of things, they are remaining present. So biochemical and industrial meaning of fermentation, if we are saying what it can be, the biochemical meaning is what? It is actually the generation of the energy. I would say that when the catabolism is there, the organic components are there, and all those organic components, they are getting catabolized, and when they are getting catabolized, they are like generating that of the energy itself. Okay, that thing we are representing it as the biochemical like mode of fermentation. So if I'm saying about biochemical mode of fermentation, what is that? We can say that it will be the production of the energy. Okay, we are getting certain kind of organic components, and whatever the organic components are remaining present, all these organic components, they are getting catabolized itself. And when they are getting catabolized, they will be releasing the energy, which is happening simply for the anaerobic respiration itself. That means for the microbes and for all these kind of the organisms, whatever it is getting happened, that we can say that the biochemical meaning of the fermentation. Now, if I'm saying about the industrial meaning, now in terms of industrial meaning, what is happening always, there is a commercialization term. That means what in this case, what will be getting happen, the production of the product by the mass culture. Okay, we are taking a microorganism, and that microorganism is being, I would say that it is being cultured in a large amount itself. Okay, in a large, huge number, the microorganisms are being cultured itself. Okay, that thing, and whenever the microbes are getting cultured, that microbes will be resulting to the production of the large amount of the secondary as well as the primary metabolites itself, and all those secondary and the primary metabolites can be utilized industrially itself. So that thing we can say that it is the industrial meaning of that of the fermentation which is remaining present. Now, if I'm saying about the range of the fermentation processes, what are the different range of the fermentation processes? The different range, the first, why the fermentation is done. That means ultimately we are tending to understand that why the fermentation is done. The fermentation is done to produce microbial cells, okay, in large number, in microbial cultures that we can see. It is done in order to produce microbial enzymes, and those microbial enzymes can be utilized in that of bakery industries, in that of medicine industries, in other food industries, and many other purposes itself. Moreover, it can be also utilized to produce out of the microbial metabolites itself. It can be also utilized to produce recombinant products. That means what? What is this recombinant product? That means a recombinant DNA technology. The recombinant DNA technology, the recombinant products, they can be utilized to that of the for that of the production also in that case. Okay. And finally, we can say that we can use that of the fermentation process in order to modify a component itself, okay, which is required to be added to that of the fermentation process itself. Okay, whenever we are taking that of a fermentation process, within that of this fermentation process, we can we can modify a compound, okay, which is to be added to that of the fermentation process itself. So for that reason also, fermentation can be utilized over there. So if I'm saying about that of the production of the microbial biomass, now when the microbial biomass, when that is getting produced, it can be divided into that of two categories itself. The microbial biomass, when it is been produced, it can be divided into two categories. What are those two categories? The first of all, obviously, production of that of the yeast itself, and the second one, it is the production of that of the microbial cells. Yeast, it will be used in that of the baking industry, and microbial cells, it is used in that of the human food or that of the animal feed industries itself. Okay. If I'm saying about microbial enzymes production, now why the microbial enzymes are getting produced? Microbial enzymes are getting produced for multiple roles itself. Here, you can see I had enlisted few of the charts which are remaining present over here, by which you can get a thorough idea that in where the microbial enzymes are getting utilized. Like in baking industry, if I'm saying, like in why the microbial enzymes are getting utilized, what kind of microbial enzyme, the amylase, the protease, both of these things are getting utilized. The fungal source as well as the fungal or bacterial source, both of these are getting utilized in the baking industry. Okay. And in the baking industry itself, dough texture, maintenance of the dough texture itself, here dough viscosity maintenance, then fermentation acceleration itself, then I would say that mixing time reduction itself, all these things they can get happen. And in order to improve the dough texture, protease enzyme is required, and in order to reduce the dough viscosity, amylase enzyme is required. Amylase enzyme extraction can be done from the fungal resources, and protease enzyme can be extracted from either fungal or the bacterial resources itself. Now, next, we can say brewing. In brewing, what can be the usage? In brewing, the mashing as well as chill proofing. Amylase and protease can be used. In cereals, the pre-cooked baby foods, where amylase are the major sources itself. The chocolate and cocoa, they can be used for manufacturing of the syrups itself in that of the amylase part. Coffee bean fermentation, or we can say that preparation of the coffee concentrates itself, that also can be utilized over here. And in confectionery, it will be used to produce sweet candies, okay, the soft center candies itself. They can be used in corn syrup, and these all enzymes, amylase, amyloglycosidase, glucose isomerase, glucose oxidase, all these different kind of enzymes, they are been like produced via fermentation itself. They are like, I would say that, and they are being utilized in that of this baking industries itself. So that is the production of the microbial enzymes and that of their advantages or that of the commercial usage. Now, if I'm saying about that of the production of the microbial metabolites, now, what are the different kind of metabolites? These are the primary metabolites which we are having: the ethanol, the organic acid, glutamic acid, lysine, nucleotides, phenylalanine, polysaccharides, vitamins. All these are what? These are all the primary metabolic products which are remaining present. Now, primary metabolites, ethanol, I would say that active ingredient that of the alcoholic beverages, we know very well, as well as the motor car fuel. Organic acids, they are used in the food industries. Glutamic acid as a flavor enhancer, lysine as the feed supplement, then flavor enhancer as nucleotides, and all these parts. So These things, all these metabolites, whatever we are seeing over here, all these metabolites, they are getting produced by that of fermentation in large scale. When they're produced in fermentation via large scale itself, they have been utilized over here itself in that case. Okay. Now we'll be seeing something about the other microbial growth and the death kinetic itself, and we'll be wanting to know that how the microbe divides. The microbe divides by binary fission, budding, and fragmentation. That is the common mode of division in which they can grow in that case. So by binary fission, single cell will be dividing into that of, I would say that the two daughter cells itself. Budding, it will be showing the formation of the single cell. A small bud will be getting produced, and that bud will be resulting to the formation of that of a newer individual itself. The fragmentation, an organism is there, and that particular organism will be producing certain kind of fragments, and those fragments that can grow and to form the newer kind of the individuals itself. Now, while fermentation, we have to know about the growth and the death kinetic itself, because in fermentation, there is always the temperature as well as pressure. These are been limited in present in the limited conditions itself. And under those limited conditions also, they are being utilized. Now, measuring the bacterial growth, there are many different methods. We had discussed these things previously also while we were discussing about microbiology part itself. Microscopic count, the plate count method, as well as number of bacteria per ml is equal to number of colonies counted on plate multiplied with that of the dilution of the other sample. Microscopic count means what? We are taking the bacterial solution and we are counting the number of the other cell under microscope. In the plate count method, we are plating the microbes and we are isolating, we are seeing the colonies of the other microbes itself. We are counting the colonies of the other microbes which is growing over there. Okay. Now, if I'm saying about the measuring the bacterial growth itself, if I'm saying about the measuring of the bacterial growth, we are having turbidimetric method. What is the turbidimetric method? It is actually, we can say that it is based on the turbidity. That means what? Like in an aqueous solution, when we are taking the microbes and those microbes which are remaining present. Now, if I'm saying about that of those microbes itself, in whatever quantity those microbes are remaining present itself, they are creating certain kind of opaqueness. That thing we are representing it as the turbidimetry itself. So we are trying to understand about the turbidimetry itself, that how much turbidness it is forming, okay, or it is showing itself, and based on that we can identify the bacterial growth. Even we can use a protein content, nitrogen content, and dry weight itself. On the basis of the same also we can identify the dry weight of the culture or the nitrogen content itself. Now, the growth cycle of the bacteria, generation time. What is generation time? The time which a bacteria is taking to complete a single division itself. That thing we are representing it as the generation time itself. Generally, in order to calculate the total number of the bacterial cells, we are having a formula as 2 to the power n multiplied with that of the n naught itself, where n naught means the initial number of the cells itself. And the number of generation, that means small n, it is having, it can be found out with that of 3.3 log to the power 10, like n itself log n naught. Like that, we can, if we are calculating, we can find out the total number of bacteria which is remaining present over here. And here we are having a growth curve. That thing we are representing it as that of a sigmoid shaped growth curve itself. Now, this sigmoid shaped growth curve, we are having the lag phase, we are having the log phase, we are having the stationary phase, and this is the declining phase or the death phase itself. In lag phase, what is getting happen whenever we are incorporating a bacterial cell over here, the organism will not be immediately showing its growth or like multiplication itself. That phase in which the microbe will be adapting itself to that of its external condition will be representing it as the lag phase. And after that, when the microbe will be like showing its fastened or accelerated mode of the division, will be representing it as the exponential phase. And after that, when the microbe will be coming to that of the stationary phase or an equilibrium phase itself, we are representing it as the stationary phase, and gradually after the stationary phase, it will be coming down. That means it will be coming down to that of the declining or that of the death phase itself. Okay. So now if I'm saying about the same, microbial killing agents. Now, what are the different microbial killing agents that are remaining present? First, heat, chemicals, and along with that, certain concentrations itself. There can be certain concentrations which are remaining present, which can kill the microbe. Okay. The thermal death time, what is happening? So thermal death time means it is actually like the time period in which the microbe or the bacteria will be taking the time to kill a specific bacterium itself. That thing we are representing it as the thermal death time. That means what, it will be always taking a specific time period in order to kill a specific bacterium at a specific temperature. We are representing it as the thermal death time. And in terms of that of calculating about the same itself, we can say like after we are studying all these part itself, we can easily identify, we can easily do that of the fermentation processes itself, when we are able to know that a microbe will be growing at what kind of the temperature, at what kind of the pressure, what kind of optimal conditions it will be requiring. If we are knowing about all these things itself, we can say that it can easily, we can reduce easily about the growth of the organisms itself. Okay. Now we have to know about the microbial food spoilage. Now we have to know about the microbial food spoilage itself. If I'm saying about the microbial food spoilage, in that case, what is getting happened? It is actually the metabolic processes itself, okay, which will be causing the foods to be undesirable, okay, or unacceptable itself, okay. And that is not undesirable, that is the undesirable and unacceptable one for the human consumption itself. Why it is undesirable and unacceptable one? Because there are certain kind of changes which are happening in the sensory characteristics itself. So when there are certain kind of sensory characteristics, that means what? Ultimately spoilage मतलब हम लोग क्या बोलते हैं, खाना जो है वो सड़ गया है. तो ये जो food spoilage, that means what? The microbe, they are able to show their growth in a faster manner within the food. And when they are showing the growth in a faster manner within the food itself, automatically they are showing their faster metabolic processes. When they are showing their faster metabolic processes, automatically the microbe will be growing in a proper manner, and it will be making the food undesirable or unacceptable for that of the human consumption itself. Human consumption, it is not possible over there. It will be showing that of the undesirable or unacceptable kind of the human consumption itself. Okay. And what is getting happened? There is a change in the sensory characteristics itself, because of which it is getting happened itself. Now, while we are discussing about the food spoilage, we have to know that what is shelf life of the food. Shelf life, this is a word, it stands for that of what? When the food, or we are coming to know that the food is durable or it is edible till what time? The food, it is maintaining its desirable qualities till what time? That thing we are representing it as the shelf life of the food itself. Okay, that means what? Shelf life of the food, it is the time during which it remains, I would say, that stable, and it retains its desired amount of the qualities itself. That thing we are representing it as the shelf life of the food itself. Okay. Now, what are the major factors which are causing the food spoilage? The first major factor, endogenous enzymes present in the plants. Second one, the insects which are infesting the food. The parasites, lots of parasites are there. Light causing degradation of the pigments, fats, and proteins itself, due to which off flavors, off odors, they can be coming. Like both of excessive heat and like physically affecting. Okay, the excessive heat can affect easily the food, its texture, and that of its flavor and other things itself. Okay, as well as if I'm talking about the moisture, if too little moisture is remaining present, that will be causing cracking, crumbling, crystallization, and excess will causes the sogginess, which will be, and even excess moisture will always be providing a better condition for the microbial growth also. So in order to preserve, or in order to prevent all the kind of these kind of the factors itself, for that of lots of kind of the food particles or food item, special processes are being launched, and all those special processes we are representing it as the hurdle technology. What is this hurdle technology? In hurdle technology, what is getting happen? It is actually an intelligent combination. That means what? All the kind of hurdles or the resistances. Okay. Microbial, I would say that, which is securing the microbial safety and stability. We are combining all these things itself. Okay. So what is getting happen? When we are considering all the hurdles, we are considering them, we are compiling them together. And when we are compiling them together, after which we are trying to maintain all the kind of parameters of the food. That means the organoleptic parameter itself, the nutritional quality parameters, and the economical viability. All these things we are considering for the food product in the hurdle technology. Now, what are the different kind of hurdles? Like temperature, water activity, competitive microorganisms, okay, like acidity. These are all that of the classical form of that of the hurdles itself. And what is the basic concept in the same itself? We are like combining existing and novel preservation techniques, which we are representing it as hurdles, in order to eliminate that of the microorganism and its growth. There are four major mechanisms by which the hurdle technology, it will be affecting the growth. The first one, homeostasis. It will be affecting the homeostasis of the microbe. It will be causing metabolic exhaustion of the microbe itself. It will be doing the stress relaxation process, and it will be showing that the multi-target preservation of the food. That means what? There are multiple targets in which the food can be preserved over here itself. So there can be three major types of that of the like hurdle technology. The first one, we are representing it as physical type of hurdle technology. The second one, we are representing it as physicochemical. The third one, we are representing it as microbial form of the other hurdle technology. Now the physical form, in physical form, what is happening? Ionizing radiation, low temperature, then high temperature, modified atmosphere, ultrasonic, all these like the physical, physiological processes have all these things. They are the types under that of the physical form of the hurdle technology. If I'm saying about the physico-chemical processes, means the kind of the processes which are involved in that of what, which are involved in that of this hurdle technology itself. If I'm saying about physico-chemical processes itself, sodium nitrate, sodium sulfide, sodium nitrite, then potassium sulfide, phosphate, salt, spices and herbs, carbon dioxide, ozone, oxygen, organic acids, all these things, these are actually the physico-chemical processes which are remaining present. Even smoking, lactoperoxide, all these things are the physico-chemical pathways itself. And we are having what we can see that we can have that of the part which we are representing in as our microbial also. We are using bacteriocins also, competitive flora we are using, and antibiotics we are using. We are combining all these hurdles, and when we are combining all these hurdles itself, we are able to create a better preservation techniques. So what are the applications? It can be used in dairy technology, okay? Like in order to enhance the shelf life in any kind of food products, hurdle technology can be used to enhance the shelf life of the product itself, okay? Even shelf stable paneer, that means what, we are enhancing the shelf life, so shelf stable paneer can be prepared. Another product, the brown peda, a traditional Indian heat desiccated milk khoa, okay, khoa kheer, kind of the other product, it can be also produced via that of this hurdle technology itself. In fruits and vegetables, there can be many kind of the other processes. We can use hurdle technology to produce the ready-to-eat foods itself. Even we can use the infrared drying as well as gamma radiation processes also, okay, which can easily reduce the microbial loads itself within that of many kind of fruits like pineapple slices and all these things itself, okay. Like fruit derived products, what can be there? The UV light, pulse light, and like they can be combined to preserve peach, pineapple, papaya, mango, and banana. All these can be preserved by using the fruit derived products itself. In meat and meat products, we can use like in order to like show the preservation of that of the shelf stable. That means what, the in order to enhance the shelf life, we can use over there itself. Okay, the proline value we can enhance, the pH, the water activity we can enhance, and all these things. So ultimately, the hurdle technology, it is used, it is combining all the resistances, all the hurdles and everything, and it is focusing towards production of that of a novel product itself. That thing we are representing it as the hurdle technology itself. Okay, students. So today we have discussed about that of the fermentation. We have discussed about that of the microbial spoilage. We have discussed about that of the food contamination and all these things, the microbial death curve, growth curve, and all these parts itself. Okay. If you're having any doubt, you can put in the chat, or you're having your class mentor available over here itself, and I'm always there. IIOCS is always there. Okay. In the next lecture, we are going to see some of the applications of that of the like food biotechnological products, like recombinant bioproducts itself, and then we'll be seeing also the important thing like the different kind of fermented foods and beverages like idli, dosa, and all these things that we'll be seeing, and we'll be wrapping up the part itself. Okay, students. Thank you, everyone. In the meanwhile, I would say that thank you, everybody. Good night. Okay, stay well, stay fit, and eat always healthy food. Thank you, everybody.